Welcome. Today we are going to learn about the structure of DNA. What do you mean by the DNA? DNA, it is a polynucleotide chain. That is, it is composed of the nucleotides, and the main function of this polynucleotide chain, it is a storage and transmission of the biological information. Here, the one more concept here, it is the gene. Gene, it is what? Gene, it is a segment of DNA molecule that contains the information required for the synthesis of biological product, and that biological product, it is what? It is a protein and the RNA. Means here the gene, it is made up of the polynucleotide chain. Okay, that is the it is made up of the DNA, and that gene, it is going to encode the proteins, or it is going to encode the RNA. Now the next one, as the DNA, it is a polynucleotide chain, you should know that what do you mean by the nucleotide, or what are the characteristic components of the nucleotides. So the characteristic components of the nucleotide, it is the nitrogenous base. Okay. Second, it is a pentose sugar. Third one, it is a phosphate. And here, the molecule without a phosphate, it is going to be called as a nucleoside. So there are the two concepts. One, it is a nucleotide, and one, it is a nucleoside. The nucleoside, it is composed of the pentose sugar and the nitrogenous base, and the nucleotide, it is composed of the nitrogenous base, pentose sugar, and the phosphate. Now going towards the next, and it is the nitrogenous bases. We know that the nitrogenous bases, they are of the two types. One, it is a purine, and the second one, it is a pyrimidine. Okay, so these purines, they are of the two types. One, it is adenine, and one, it is a guanine. Now, when you look at this, okay, when you look at the structure of this adenine, at the sixth position, there is what? There is a presence of this NH2. Okay, and here, when you look at the structure of the guanine, at the sixth position, there is a C double bond O, and at the second position, there is a presence of the NH2, and at this, okay, so in this way, there is what? There is a structure of the adenine and the guanine, which has been derived from the parental compound, and that parental compound, it is a purines. Second one, it is a pyrimidine. Okay, the pyrimidine, they are of the three types. One, it is a cytosine. Second, it is a thiamine, and third, it is a uracil. Now, this cytosine and the thiamine, it is present into the DNA, and the cytosine and the uracil, it is present into the RNA. That is the basic difference between the DNA and RNA. According to the basis, it is the presence of the uracil in the RNA and the presence of the thiamine into the DNA. Now, when you look at the structures, okay, when you look at the structures of the cytosine, it has the NH2 group at its fourth position. The thymine, it has what? Thymine, it has the C double bond O at the second position. At the fourth position, there is a C double bond O. So when you look at the structure of the cytosine and the thymine, here at the fourth position, cytosine has NH2, and thymine, it has the C double bond O. The second position, it is the same one, C double bond O, C double bond O. But here, okay, at the fifth position, what you observe that there is what? There is a CH3, and that CH3, it is not present into the cytosine. Okay. Now, when you look at the structure of the uracil, okay, when you look at the structure of the uracil, here at the second position, there is a C double bond O, which is similar to all types of the pyrimidines. But at the fourth position, there is what? There is a C double bond O. Okay, like whom? Like the thymine. But what is the difference then? At the fifth position, there is a CH. Okay, now when you look at the structure of the thymine and the uracil, okay, when you look at the structure of the thymine and uracil, what you find the difference? The difference, it is what? At the fifth position, there is a CH3 in the thymine, and there is no presence of the CH3 into the uracil. So this, it is a basic difference between whom? Between the thymine and the uracil. Okay, now going towards the next one. Here it is a pentose sugar. Now, the deoxyribose, it is particularly derived from the ribose sugar. When you look at the ribose sugar, at the second position, there is a OH. Okay. And when you look at the deoxyribose sugar, at the second position, there is no presence of the OH. That is, there is the removal of O from the second position, that is at the 2'. So the ribose, it has OH at the 2' position, and in deoxyribose, there is no presence of the O at the 2' position. So this it is a second difference between the DNA and the RNA, that the RNA it is consisting of the ribose and the DNA it is consisting of the deoxyribose. Now here it is an phosphoric acid from where the phosphate group it is going to be taken up. Now when there is a synthesis of the nucleotide, the sugar, okay, deoxyribose sugar, okay, it is going to interact with the base, okay, and there is a bond formation, okay, there is a bond formation between the first prime, that is a 1 prime, and the N9 position of 19 position of the purines, okay, and here you can see here, okay, you can see here there is a removal of one H2O, and now the first molecule which is going to be formed, it is whom? It is a nucleoside. As the deoxyribose, it is getting bound with the base. Now the next one, it is going to come the phosphoric acid. Okay. Now here the phosphoric acid, the interaction it is between the 5 prime position of the deoxyribose, and here the interaction it is in between the phosphoric acid. So here also there is what? There is a removal of one of the H2O, one of the water molecule, it is going to be removed. Okay, and now here this phosphate group, now it is going to be attached to who? Attached to the deoxyribose, forming the ester bond. Okay, this it is an ester bond, and here it is what? It is the bond between the deoxyribose and your base. It is what? It is a glycosidic linkage. Okay, or the glycosidic bond. Okay, so in this way there is the synthesis of, or there is a formation of your nucleotide. Okay, now the next one here, when you look at the structure of the DNA, the first of all there is the incoming of the Chargaff rule. Okay, and the clue for the structure of the DNA, it has been given by the scientist Chargaff in 1940. And what he has said that, the base composition of DNA generally varies from one species to the another. That is the amount of the DNA which is present into the species. Okay, the amount of the nucleotides which are present into the species, they are going to differ from species to species. And here next one, it is what? DNA specimen isolated from the different tissues of the same species. Okay, means what? Of the same organism. Okay, same organism, or suppose you are taking one animal, like suppose you have taken the rabbit, and from the rabbit you are taking the sample of the DNA from its lungs or heart. If that sample it is consisting of the same amount of it is going to consisting of the same amount of the DNA. Okay, and okay, in this way the DNA specimen isolated from the different tissues of the same species have the same base composition. Okay, the base composition of the DNA in a given species does not change with the organism's age. Suppose an organism it is there. Now the organism it is of the 1 year, or it is of the 12 year, or it is of the 30 years. Okay, the base composition it is not going to change. That is, it is going to be a constant. Now, last one, it is a in all cellular DNAs, regardless of the species, the number of adenosine residues is equal to the number of thymine residues. That is, okay, that is A plus G is equal to C plus T. Okay, that is the amount of purines is equal to the amount of pyrimidines. Okay, this it is called as the Chargaff rule. Now going towards the structure of DNA. Okay, the clue it has been given by the Chargaff, but next the structure of the DNA it has to be elucidated, and for that purpose the X-ray diffraction it has been done for the DNA fiber, and it has been done by the Franklin and the Wilkins. Okay, Franklin and Wilkins, they has done the X-ray diffraction of the DNA, and they have got the characteristic X-ray diffraction pattern. And what they have deduced, okay, what they have deduced, that the DNA molecule are the helical with two periodicities along their long axis, and it has one primary one, it is a 3.4 angstrom, and a secondary one, it is a 34 angstrom. but they cannot give the three-dimensional structure, and that three-dimensional structure of the DNA has been given in 1953 by the Watson and Crick, and they got the Nobel Prize for it. Now here there is a structure of the DNA. Now it is what the when you look at the structure of the DNA, it is a double helix. Okay, this one. Okay, this one it is a one helix. Okay, and the second one it is a this one helix, and these two helices they are anti-parallel to each other. Now here it is an one of the helix, here it is a 3 prime and another it has a 5 prime, that is a 3 prime OH and a 5 prime phosphate it is present, that the nature it is a anti-parallel. Okay, and here in one, okay, this it is one helix. Okay, this one it is a one helix, and this one helix it is consisting of the base pairs, and that base pairs it is about the 10.5 base pairs, and this helix it is of the 34 angstrom. Okay, and the rise per, okay, that is the rise per turn it is the 34 angstrom, and the distance between these two bases it is the 3.4 angstrom. The diameter of the helix, okay, the diameter of the helix it is a 20 angstrom, that is a 2 nanometer. And when you look at this structure, okay, when you look at this structure, what you observe that backbone, okay, at the backbone of the DNA, who is there? There is a presence of the sugar phosphate. That is the hydrophilic group. It is present outer side, and internally, okay, internally in between the two helices, there is what? There is a presence of the bases. Okay, these are the bases which are present, and where they are at internally, and these bases when they are internally present they are forming the hydrogen bond. This you can see these red lines, these are what? These are the hydrogen bonds. That is the bases, okay, bases, two bases from the two helices, they are going to interact to each other with the help of the bond, and that bond it is what? That bond it is a hydrogen bond. That's why the two DNA molecules, they are going to be held together. Second interaction to stabilize the structure, it is the interaction in between the bases, and that interaction, it is a hydrophobic interaction. That is, there is a stacking interaction. Stacking means what? Stacking of the bases, it is there. That is stacking of the bases, like how the coins, they are stacked one above the another. That it is what? That it is a stacking interaction. It is going to be there. Okay, in this way, this it is what? This it is a structure of DNA. Okay, now going to the next one, the DNA. It may be the right-handed or it may be the left-handed, but often, okay, oftenly or at the most, the structure which is present, it is a right-handed double helix. Now here you can see the base pairing, okay, polynucleotide polymer it is there, okay, and this polymer, this internally, okay, this structure, okay, it has been given here, okay. So here it is an adenine, it is going to binding with the thymine, okay. Now here this it is a first, second, third, fourth, fifth, and the sixth position. At the sixth position, here it is the NH2 into the adenine, and this NH2 it is involved into the hydrogen bonding with whom? With the thymine, okay. And here the position it is the 1, 2, 3, 4. Fourth position of the thymine, and fourth position of the adenine, and the fifth position, first position of the adenine, and here third position of the thymine, it is going to form the hydrogen bond. So there are the presence of the two hydrogen bond, and here also you can see in the guanine and the cytosine also there is a presence of what? There is a presence of the hydrogen bonds. Okay. Now here DNA, in the DNA and RNA, the nucleotides they are bound to each other with the help of the bond, and that bond it is called as an phosphodiester linkage. Okay, there is a ester linkages. There are the two ester linkages. This one and this one. These are what? These are the ester linkages which are present. That's why it is a phosphodiester linkage. Okay, it is a phosphodiester linkage, and there is a phosphate group it is present. That's why it is a phosphodiester. Okay, Now the next one, forms of the DNA. Okay, there are the three forms of the DNA: A form, B form, and the Z form. Most of the time, okay, the most of the time the DNA it is present into the B form, and the helical sense into the B form of the DNA. Okay, the helical sense of the B form of the DNA, it is a right-handed. Diameter it is a 20 angstrom. Base pair per helix it is a 10.5, that is per turn there is the 10.5. Rise per helix, that is the distance between the two bases, it is 3.4 angstrom. Okay, then helix axis it is a 6 degrees, and a sugar pucker conformation it is a C2 endo, and glycosyl bond conformation it is an anti in the B form of the DNA. And this B form of the DNA it is the most frequently observed into where? It is most frequently observed into the DNA. Second form, here it is an A form. A form it is also a right-handed. Okay, diameter it is a 26. Okay, the base per helix it is a 11. Okay, rise per helix it is a 2.6 angstrom. Helix axis. Okay, base tilt normal to the helix axis, that is a tilting. Okay, it is of about how many? It is of about the 20 angstrom. Sugar pucker conformation it is a 3 prime C3 prime endo, and a glycosyl bond conformation it is an anti. The third form it is a Z form. Okay, when the salt it is a Z form of the DNA. It is going to be observed into, most of the time it is observed into the regulatory sequences. Okay, and this Z form it is a left-handed. Okay, diameter it is 18 angstrom, and base pairs per helical turn it is a 12. Okay, helix rise per base pair it is a 3.4. Base tilt normal to the helix axis it is a 7 degrees. Sugar pucker conformation it is a C2 endo for the pyrimidines, and C3 prime endo for the purines. Okay, glycosyl bond conformation it is a anti for the pyrimidines, and a cis for the purines. Okay, so this it is regarding the structure of DNA. Now going towards the questions. Okay. Now, which position in the purine ring of the purine nucleotide in the DNA have the potential to form the hydrogen bonds but are not involved into the Watson and Crick base pairing? Okay. The question it is requiring the answer in the single or the single word. Okay, and it is a N3 and N7 position. Now here when you look at the structure of this adenine and guanine, okay, this one, okay, this it is a first position, second, third, fourth, fifth, and here it is an sixth. Now here it is an third position. Okay, this it is a third position, and this it is a seventh position of the adenine. This it is a third position of whom? Third position of the guanine, and here it is a seventh position of the guanine. Okay, now these two positions, okay, though they are able to form the hydrogen bond, they are not involved into the hydrogen bond formation. Now, here you can see, okay, what you can see here, that is your first position, though it is an N, it is able to form the hydrogen bond. The same type it is present into here at the third position and at this seventh position. So that is this N and this N, the N at the N3 position and the N at the 7th position, it is able to form the hydrogen bond, but they are not involved into the hydrogen bond formation. Now the second question, it is what? One strand of the double helical DNA. Okay, it has a sequence. Okay, the sequence it is what? G C G C A A T A triple T C T C A T A T T G C G C. That is such a type of a sequence it has been given. Now write the base sequence of the complementary strand. Okay. What special type of a sequence is contained in this DNA segment? Does the double-stranded DNA have the potential to form any alternative structure? Now, when you look at the when you look at the sequence, it is going to form the alternative structure. Okay, and that alternative structure, it may be a hairpin or it may be a cruciform. Now, here when you look at the sequence now, okay, when you look at the sequence of it, it is inverted repeat. Okay, inverted repeat, it is like how here you can see this one GCGC at 5 prime, and at 3 prime it is what? It is a CGCG. It is a inverted sequence. Okay, and when you look at the next sequence, it is a AATA TTT, and here you can see TTATAA. Okay, so the sequence, it is how? The sequence, it is a inverted repeat. Okay, the sequence, it is a inverted repeat. And now this G, okay, the G at this 5 prime and the, okay, G at GC at this 5 prime, it can complementary base pair with whom? The CG of this 3 prime. Okay, and here next one, it is whom? It is a AAT ATTT. It can complementary base pair with whom? TTA T. Okay, then here AA. Okay, so here when such a type of an complementary base pairing it is going to be there, at that time it may form the hairpin or it may form the cruciform structure. Okay, got this. Now the next one, hairpins may form at the palindromic sequences in the single strands of either of the DNA or the RNA. How is the helical structure of the long and the fully base pair except at the end hairpin in RNA different from that of the similar hairpin in the DNA? Now, what is asked now? First, understand what is asked. Now, here it is a inverted repeat, and this inverted repeat it is going to form the hairpin structure. Okay. Now, such a what it has been given into the question, that there is a palindromic sequence. Okay, that is read from any one end. It is going to be a same one. Okay, from the 5 prime to 3 prime, 3 prime to 5 prime on the two strands of the DNA, it is the same sequence, that it is a palindromic sequence. Now, here what has been said, that their sequences, they are self-complementary base pairing and it is forming a secondary structure. The RNA, it is the single stranded RNA, it is going to form the secondary structure, and if the DNA it is in a single stranded form, here it has been said that it is going to form the hairpin structure. Now both of the structures, they are how? Both of the structures, they are the double stranded. Okay, they are the double stranded as they are bound to each other. Now, just now we have learned into the structure of DNA, forms of the DNA. Okay, what we have learned, the forms of the DNA, that the B form of the DNA, it is a common into the DNA, and the A form, it is common into the RNA. So this, it is a basic difference between the DNA and RNA. When the DNA it is going to form the double stranded structure, it is going to form the B form of the DNA, and when the RNA it is there, the RNA it is going to form the A form of the DNA. So here the distinguishing character, it is what? Distinguishing character, it is that you have to find out that what form of the DNA it is present, or the what form it is present, A form or the B form. If it is A form, it is an RNA. If it is a B form, then it is going to be a DNA. Now explain why absorption of the UV light by the double-stranded DNA increases hyperchromic effect. That is, the absorption it is going to increase when the DNA it is denatured. Now you have learned the structure of the purines and pyrimidines. In the purines and pyrimidines, okay, the double bonds resonance it is there, and that resonance into the double strand, resonance into the double bond, it is responsible for absorption of the UV radiation. Okay, now in the DNA, okay, when it is a double-stranded DNA, okay, the bases they are placed internally. Okay, that is the UV light which is going to get hitted, or the UV light which is going to come in contact with the bases, it is a lesser when it is in a double-stranded form. But as there is melting of the DNA, the bases they are going to be exposed more, and as it is going to be exposed more, the more of the purines on the pyramids, they are getting exposed to the UV radiation, and then they are going to increase the absorption. Okay, so here Purines and pyrimidines are responsible for the absorption of UV radiation, and as the radiation it is going to be increased, the exposure of the purines and pyrimidines to the UV radiation it is going to increase, and thus, thus you are going to observe there is a hyperchromic effect. Now the next one, it is in a sample of the DNA, in a sample of the DNA isolated from the unidentified species of the bacteria X and Y. That is, isolation of DNA has been done, from the two bacteria X and Y. Adenine makes up to 32% and the 17% respectively. That is, the X has the adenine content 32% and the Y has adenine content 17%. From the total basis. What relative proportion of the adenine, guanine, thymine, and cytosine would you expect to find in the two DNA samples? What assumptions have you made? One of the species was isolated from the hot spring, 64 degrees Celsius, suggests which species is a thermophilic bacteria. What is the basis of your? So here there are the two bacteria, they are isolated. One it is a X, one it is a Y. The Y bacteria, it is a thermophilic. Now here, Y bacteria, it has a 70% adenine, thus have the 83% of the guanine. That is, it has a high GC content than the 80, which is a characteristic of the thermophilic bacteria. Now, from the 100%, there is a 17% adenine. So you have to subtract the 17 from 100, and it is going to be a 83%. Same you have to do for whom? Same you have to do for the X species. The X species, it has the 32%. X species has the 32% of the adenine. Means means here there is though there is a GC content high. Though 32%, 80 it is there, means remaining it is a GC. But when you look at this, when you look at these percentages which has been given, the Y bacteria, it has a more GC content than whom? It has a more GC content than the X. Now, when the bacteria it is a thermophilic, means that bacteria it is living in the that bacteria it is living in where? In the high temperatures. So to stabilize the DNA at a high temperature, it is requiring the more of the GC content, because to melt the GC, to melt the GC, that is to melt the G triple bond C, the higher energy it is required. So as the GC content it is a higher one, the melting of DNA it is a difficult one. That's why in the thermophilic bacteria, the GC content it is going to be a higher one. And here when you compare these two, the GC content of the Y bacteria it is a more than the GC content of the X bacteria. That's why the that's why the Y bacteria it is what? The Y bacteria it is the thermophilic bacteria. I think you may have cleared the idea of the DNA structure and some of the questions regarding the structure of DNA. If you like my video, please subscribe my channel and also share my video with your friends. Thank you.